Hi students, welcome to HSC Biology and Module 7, Infectious Disease. This is video number 22 and we're going to have a bit of a look at antimicrobials and in particular antivirals. So what we want you to do in this particular task is to investigate and assess the effectiveness of pharmaceuticals as treatment strategies for the control of infectious disease. And in this one, we're going to focus on antivirals. To give yourself a scale of things that you need to be able to do, the first thing you must be able to do, obviously, is to define the term antiviral so we know what we're talking about. We'd like you to be able to describe a range of chemotherapies used to treat infectious diseases and perhaps to be able to discuss why specific pharmaceuticals are needed for specific groups of pathogens. So in our last series, we were looking at some of the different ways that we have to try and limit the spread of infectious disease. Now we're going to look more at some of the treatment methods for how we can help or give our body assistance when we do develop an infection. We know now that there's both a chemical and biological response and a combination of physical and chemical barriers that help protect us against pathogens. But none of these are 100% foolproof. None of these are 100% guaranteed to keep us from any possible infections. And sometimes we still contract infectious pathogens. And as a result, we need to be able to treat those, particularly some of the more serious ones that we may contract. We know that there's a range of different types of pharmaceuticals that can be used, chemicals or drugs that have been specifically made for specific purposes, in the same way as our immune system works. Now, the term chemotherapy often is associated with cancer treatment, but chemotherapy is literally chemicals for therapy. So any chemicals that have been produced that are used for medicinal purposes. And so therefore we can use chemotherapy to treat human disease or disorder. When we're specifically looking at infectious disease, then we're trying to combat microorganisms that have penetrated through those first few lines of defense. They've got past our physical and chemical barriers. They've been able to hide long enough not to be caught by phagocytes, and so they are causing some damage. And while our system is learning, while our third line of defense is learning what type of pathogen, what type of antigens, and what specific type of response our body needs, sometimes we need a little bit of assistance in this fight. Unfortunately, there's no one chemical that just seems to combat everything or give our system a sufficiently high enough boost that we don't need anything else. So this class of chemotherapy drugs or antimicrobials is subdivided into the specific types of chemicals that attack certain types or certain groups of pathogens. So most of them are pretty obvious from their name. An antiviral is something that combats viruses. The antibiotics are probably the ones you're most familiar with, and they specifically work on bacteria, which is why if you've got a cold or a flu, antibiotics are not going to help, not unless you've got a secondary infection, because they target bacteria, they don't target viruses. Antifungals are ones that target particular types of fungi, and antiprotozoals, again, well, you know, the name I think pretty much suggests exactly what the function is. In this particular video, we're going to focus on just the antivirals, and we will devote a separate video to the antibiotics. Now, antivirals are relatively recent, about the 1960s. This is about as long as we've had antivirals, and we needed something that, because the antibiotics were not working on viruses, so they were fantastic in combating bacterial infection, but no good for viruses. So we needed something specific to target viruses. So things like Tamiflu. Now Tamiflu is kind of the package name. The specific antiviral agent that's in that particular product is oseltamivir. And that is the specific antiviral that works against at least one of or a couple of the strains of influenza. Now, we'll talk later on about the mutation rates and why certain types of disease-causing pathogens are continuing to resist our best attempts to try and kill them off one way or another. But nevertheless, Tamiflu is one of these antiviral products that we can take if we contract influenza. What they do is they slow the growth of the virus. So they don't always kill it, but what they do is, if we can understand the growth phase of viruses, we know that they don't tend to survive for long periods outside of cells. They need to be inside of host cells. They certainly, if they're going to reproduce, need to be inside of host cells. So if we can trap them, stop them from entering host cells, or if we can trap them inside a host cell so they can't burst back out, then there are ways there that we can kind of slow down the growth of the virus and give our actual immune system time to develop a specific response. The earlier you take an antiviral, the more likely you are to minimize the symptoms. So the plan is that if you know that you've contracted one of these, then you try and get to the course of antivirals as quickly as you possibly can. Now, antivirals tend to do one of three main things. They either target the capsid proteins, which is usually the little protein that's around the genetic material. Now, in viruses, that could be DNA or RNA. And there'll be a single or double strand of genetic material. And usually that's protected by a protein capsid, like a little cap around that material. Around the virus itself, there can be a protein, a sort of encasing, if you like, around the whole of the virus. And often as it's emerging from a host cell, we get these little envelope proteins, and antivirus can actually attack these as well. Or we know that the main way that viruses are going to reproduce is through the replication of their DNA or RNA. And so that's going to involve polymerase enzymes, again, a type of protein, and these antivirals are going to perhaps disrupt the function of the polymerase enzymes and stop the replication process from occurring. So there's a number of different ways in which antivirals can work. What's their effect? Well, what they do is one of a couple of things, and I've kind of briefly talked about this, but let's have a look at each of these ones. And in the background of this slide, I've just put in a kind of slightly enlarged version of a virus, and you can see the envelope proteins. You can see the capsid, the nucleocapsid, where the genetic material is sitting inside of the virus. So you get a bit of a sense of where these things are. So we stop them from getting into the host cells by binding to receptors, or we inhibit the enzymes needed for reproduction, or we block protein synthesis of important viral proteins, so we actually stop them from producing or expressing the genes that are part of their genome, or we isolate the virus from within the host cell. So this is not in, and this is not out. So if we can stop them getting in, that's great. If they get in, they're going to reproduce. So we're going to end up with multiple copies of the virus. So if they do, if we can stop them from getting into the host cells, that's great. If we can't, maybe what we can do is we can stop them from getting back out of those cells. If we can't do either of those things, then maybe we can just stop the replication processes. So these are the four main ways in which antivirals work. And they are one class of antimicrobials or chemotherapy treatments for infectious disease. Another one, probably better known, is the use of antibiotics. But we'll look at those in the next video. Thanks for watching.